Okay, so first let me thank the organizers for inviting me to this conference. What I want to do is tell you of some of the work we've been doing recently at Institut Curie on tissues. Most of what I will say will be theory. However, there is a small experimental part to show you what kind of experiments we're trying to do. The theory part is the work of two PhD students, Jonas Ramp, that we share with Frank Kulischer in Dresden, and Edouard Ranzo, and there is always Jacques Pro in all these kind of stories. The experimental part is done at Institut Curie, not by me, because I don't do any experiments, by Morgane Delarue and Fabien Montel. So my talk will have two parts. In the first part, what I want to do is try to show you how a tissue invades another tissue. And before I do that, I briefly try to convince you that if you look at very long time scales, days, you can treat tissues as liquid materials. And then I'll try to tell you our story about tissue invasion. The second part of my talk will have to do with the intestine and the structure of the intestine. There is a structure in protrusion called villi that we've been interested in. I will first do some kind of mechanical analysis. And what we tried to do recently is to couple this mechanical analysis to cell differentiation. If there is time, I'll tell you about tubular structures. So let me start by tissues. The tissues that we look at, mostly experimentally, are these cell aggregates, which are called spheroids. The way they are made is they aggregate a few cells, and then they put it in a medium. The cells divide, and they grow as spherical objects like this, which are called spheroids. Takes a few days, cell division in 1 day. So typically, you wait 10 days, 20 days. And after 10 or 20 days, you get an aggregate like this. It grows, and then it stops growing. And the size is a few hundred microns, 500 microns or something like this. So these are various kind of cells and various kind of ways of imagining them. These pictures come from a view of Mr. Stelzer. The first thing you can notice on these tissues is that all these aggregates more or less look like spheres. And this has been known for a long time. And this gentleman here, Malcolm Steinberg, in the 70s proposed that there is something like an interfacial tension, isotropic interfacial tension. His idea was that this was kind of like a liquid drop. And if you want essentially to balance the forces with the interfacial tension, it should look like a sphere. So it's a sphere plus a few fluctuations. Sometimes it's a little bit deformed. Now, why would it be like this? The naive idea is that cell adheres to each other. So a cell which is on the surface, it's like the old ideas on surface tension by Laplace and people like this, or Young. On the surface, it has less neighbors, so it has less adhesion energy, and it costs energy to put a cell on the surface. The idea is kind of okay. The interpretation in terms of adhesion molecules is not sufficient. In particular, it's now well established that things like actin and myosin have an important role on this surface tension. So that's the first idea on why a tissue should look at a liquid. The other part of it is mechanical properties. Now, if you take an aggregate like this and you push on it, at very early times after you push on it, it looks like a solid, and what you measure is more or less the elasticity of the cells. So the cells have a shear modulus, which is very low. It's like 1,000 Pascal or 100 Pascal in certain cases. But it's finite. And in early stages, it's not exactly the elastic modulus of the cells, but it's close to the elastic modulus of the cells. The second point that I want to mention is that this elastic stress is not the only stress in the tissue, because cells divide and cells die. Now, if a cell wants to divide, it has to push the neighbors because it needs room. So a dividing cell exerts not a force, because the total force should be 0. So the simplest thing it can do is exert a force dipole that pushes the other cells outwards. Similarly, a cell that dies exerts a force dipole that pushes the other cells inwards. Now, if you sum all of these dipoles, you have some internal stress in the tissue, and this internal stress varies with time because you have continuous cell division and cell death in the tissue. Now, there's an experimental fact that you have to keep in mind, which is that if a cell is under stress, the axis of cell division is related to the axis of the stress. There is a very nice experiment at Institut Curie, which is due to Mathieu Piel, where they take an elastic sheet, they put a cell on it, and they pull on the elastic sheet. And in all cases, the cell divides along the direction where you push. What this tells you is that the axis of cell division is the principal axis of the stress. Now, if you put all of this together, and I don't want to go into the detail of that, what this tells you is that in a tissue, the stress can relax, and it can relax because of cell division and cell death. Yes?
But cell division itself does not change the volume, right? So isn't it rather growth?
So when a cell wants to divide, it grows, and then it divides. So what I'm calling cell division kind of includes the growth and the division, which means that everything I will say has no value if you go at very short time scale below the cell division time. So the time scales that I will talk about during all my talk, something like 1 day or 2 days or 20 days in certain cases. So cell division and cell death can relax the stress. So it means that a tissue for me is a viscoelastic material, like a Maxwell material, with a relaxation time which is 1 over the rate of cell division. Or if you want that the viscosity of the tissue is just the elastic modulus of the cell divided by the cell division rate. So that's kind of the two elements that I have to make me think that if I look at very long times, a spheroid like this more or less behaves as a liquid. Now I can be a little bit more quantitative. The way you introduce cell division rate and cell death rate is by writing conservation laws. So this is the flux of cells. This is the convection of cells by the velocity. And this is not conserved because cells divide. And cells divide with a rate, which is the inverse of the time between two divisions. And they die, and the death of cells in these aggregates is due to what's called apoptosis, which is programmed cell death. If you write it like this, it looks pretty simple. However, all the biology is hidden into these two parameters here. In particular, I put here that they would depend on the density, they depend on pressure as well, but they depend on all kind of biochemical parameters. So what I will do is which is this, what I call here the biochemical state. I will assume throughout that this biochemical state remains constant, so that I have a way to maintain constant the density of nutrients, the density of growth factors, all these chemicals which are important and without which the cell can die. They are included in these factors here. Now, the cell division rate depends on density. You could also say that it depends on the pressure. Now, there is a distinction that should be made between two pressures. One of the pressures that everybody would think of is the hydrostatic pressure. Now, this is not the one I have in mind. The one I have in mind is the pressure exerted by the deformation of the cell, something like the cell pressure. Now, there are lots of experiments, and most of them agree that the cell division rate decreases with pressure, and the cell death rate increases with pressure. If you have very few cells, they are happy and they divide freely. And then if you compress them and compress them, you decrease the division rate, you increase the death rate, and you reach a pressure where they become equal. Now at this pressure, you have as many cell division as cell death. So cell division on average compensates cell death, and the tissue is at a steady state. So this pressure is what I'm going to call the homeostatic pressure of the tissue. Now in the following, I won't consider too much tissues exactly at this homeostatic pressure, but I will consider tissue in the vicinity of this homeostatic pressure.
So,
Wait.
if you kill a cell, what happens to its mass?
So that's a good question. Yes, so there are two answers to that. I get this question all the time. So if the cells die, there are debris of cells. In vivo, that's no problem because you have macrophages and cells like this that clean it out. In vitro, we don't have that because we know exactly which cells are there, and there are no cells like this. Part of it is eaten by the neighboring cells, and part of it remains in the tissue. If you wait for a very long time, This is why the experiments that we show you stop after 15 days or so. What happens is that the core of this spheroid, there is what people call a necrotic core. So all the dead material accumulates in the center. It's not quite true if you do the experiments on shorter time scales, so I will stop the description of what happens before 15 days.
I was wondering if you could more generally, P is the trace or a fraction or multiple of the trace of the stress tensor. Could you have the growth and death rate depend on, say, the eigenvalues of the stress tensor?
Yeah, so some tissues are polarized, which means that on average, the cells divide along a direction. It's true in development, for instance, because you have chemical signals that force the direction of cell division. Then it's like an anisotropic medium, and you want the cell division and cell rate depend on the whole stress tensor. I won't talk too much about that, but there's another thing that can happen, is that the tissue spontaneously polarizes. And in fact, if you go through the derivations that it didn't give you, there is a case where you see spontaneous polarization of the tissue without any external signal. So it has, in a sense, nematic order, where the nematic direction would be the direction of cell division. And in this case, of course, my description in terms of isotropic stuff or isotropic stressors is not sufficient to describe what I want.
Okay, so the more general model wouldn't do that.
A more general model, and I have to refer to that because I told you that the whole story of liquids is that the stress orients the cells or orients the cell division. So in the middle of this derivation, I have an order parameter, which is the axis of cell division. If you're in a disordered state, in a sense, it relaxes to zero with time, and it doesn't matter. If you reach a state where the cells wants to be ordered, there is a kind of dynamic phase transition, which corresponds to the polarization of the tissue. We haven't studied that too much.
Question related to Tom's, which is when you say pressure, it's of course not hydrostatic pressure, but it's pressure keeping what?
So I'll show you.
Constant,
I'll show you.
What is the pressure exerting on it?
So let me show you the experiments, and then if you're not happy, you can ask the question back. So the experiments, one of the things we wanted to do is measure this pressure. Now, at the back of our mind, this is an information on the behavior of the cells, and maybe you get, these are mostly cancer cells, and you might get information from that on how invasive they are, and it's not harder to measure that than to measure any biochemical or chemical parameters. So that's what we had in mind. So they take, the way the experiment is done, they take one of these spheroids. You see this one is something like 100 microns, and here they follow it 10 days. Now it's been done with 5 or 6 cell lines. They are always cancer cells. The reason is a technical question. It's very hard to make an aggregate like this with a normal cell. Cancer cells divide without asking any questions, so they grow easily, and it's very easy to make that. The only thing you need to avoid is infections by whatever. Then what they do is they have everything that the cells need here to divide, like nutrients, growth factor, and these diffuse in and out the aggregate. The way we apply a force on this, or a pressure on this, is we put a polymer in the solution outside. It's a sugar polymer called dextran. It's very long. Now, if you label this polymer, what you realize is that it never goes inside the aggregate. So in a sense, the surface of the aggregate, because of the tight junctions between the cells, acts like a semi-permeable membrane. So what you create is an osmotic pressure difference between inside and outside. And this osmotic pressure difference, when the stuff equilibrates, creates a pressure. So you have a force pushing on the aggregate, but it's not the hydrostatic pressure because water goes in and out freely. So it's something intermediate between a pressure and an osmotic pressure.
It's osmotic only with respect to dextran or something like
It's osmotic only with, yeah, because you need the nutrients inside. If you don't have any nutrients, everybody will die. So I'll take any of these. The one that we studied a lot is this one. At zero pressure, you follow this curve here. So you measure the volume, which is normalized in a certain way. It grows with time, over here 10 days, and after 15 days or so, it saturates to a finite value. So typically, the finite value would be slightly less than a millimeter. And then you increase the pressure, it grows less, and the finite radius is smaller. And that's true for all of these cases. Now, orders of magnitude of the pressure is like a few thousand pascals. And this curve here, where it essentially doesn't grow, are something like 5,000 pascals. Now, there's another experiment you can make. You can take a cell, and you apply a huge pressure on the cell, and you see whether it survives or not. If you go to atmospheres or tens of atmospheres, the cells die. But these pressures are much, much higher than the one I'm talking about. Now, wait.
Is the rate of division constant inside the aggregate?
The rate, we assume, it seems to be reasonably constant. I'll show you, this is what's on the next slide. So biologists can label everything. In particular, they can label cells that divide. So this is the ones which are labeled in blue. And cells that die, which are the ones labeled in red. Now, you have to be careful that the blue ones are dividing. The red one is cumulative. It's all the cells that have died. This half of the drawing is made without external applied pressure. This part is made at a pressure of 1 kilopascal. Now, what's very clear here is that there is an external layer of cells where cells divide more. So cells divide on the surface. In the bulk, cells mostly die. So cells disappear in the bulk and divide on the surface. Now, if you take the ratio between the surface and the bulk, it gives you a length scale, and that's the final size of the aggregate. So we fitted the curve that I showed you before with an extremely naive model. We assume that the cells are incompressible, so the number is the same as the volume. So the total volume is the cells that disappear throughout the bulk, and they appear on the surface. So the term on the surface is proportional to the area, which is the volume to the 2/3 power. Then you solve this equation, and you fit the curve. And you have two parameters to fit, which is the bulk growth rate, which is negative, and the surface growth rate, which is positive. You extract these two parameters, and you can plot them as a function of pressure. The surface growth rate doesn't change very much, and the negative growth rate decreases strongly with pressure, which is what I told you before. If you increase pressure, cells have a tendency to die, and they have a tendency to divide less. Now, you can look more carefully, and in fact, the quantity that changes is the cell division rate. Cell division decreases a lot when you increase the pressure. Now, why does it do that? There are two naive reasons that you can think of. The first one is nutrient effects. The nutrients are outside here. They diffuse, but they are eaten by the cells which are here, so the concentration of nutrients decays exponentially. So if the cells on the surface have more nutrients, they will divide more, and this is why you have this layer here. There is another effect, which is kind of a crowding effect. What I told you is that when a cell divides, it needs to exert forces on the neighboring cells. There are less neighbors on the surface, so because there are less neighbors, it's easier to divide on the surface. So they try to minimize the nutrient effect. I cannot swear that in all of the experiments, nutrients don't count. So maybe part of what you see is due to nutrients. We believe that there is some crowding effect. Now,
If things are dying inside faster than they're being born, there has to be a flow, right?
that's what I'm going to show you. So as John was saying, cells divide on the surface, and if you reach a steady state, they have to move inwards. So the way the experiment is done is you grow a spherical aggregate, and you reach steady state. When you're at steady state, you inject fluorescent particles on the surface. So the initial edge of the aggregate is here. At t equals 0, you inject particles on the surface, and then they are carried inside by the flow. And after 4 days, there are the green particles which are here. So you can measure the density, and you see that the peak of the particles moves inwards. You can do that at 0 pressure. You can do that at finite pressure. Sorry? So it spreads because the density is not constant. The density profile is plotted here. You can imagine diffusion. There is a diffusion of cells because there is at least noise associated to cell division and cell death that we treat as random processes. It seems, and we did numerical simulations with diffusion and things, that diffusion doesn't play much role. So what you see here is just the transport inside by the flow, plus the broadening, which is due to the fact that the velocity is not constant. And if you assume that, it fits extremely well. Now, there's another way to do the experiment. You inject the particles, but you inject the particle before the aggregate reaches steady state. Then there is a growth. So the growth will take the particles outwards compared to their initial position. So at short times, they go out, and at long times, they go in. And all this fits very well, so we're pretty confident that there is a flow, and it's exactly what you can guess. And we can measure the growth rate and the cell division rates from these pictures, and they fit pretty well with the one we derived from the previous experiment. Now, then, of course, there's a big question, is why does pressure affect cell division and cell death? The real answer is I don't know. But they tried to do experiments, and I'll tell you what the experiments give. So the way the experiment is done is they grow the aggregate for 4 days, and then at 4 days, they apply a pressure. And then they look at what happens. So they looked at 3 things. They look at the volume of the cells, and it's not exactly the volume, it's the distance between nuclei that they look at, and they assume that it's close to the volume. They look at a protein which is called P27, which for the expert is a kinase, because it's been known that this protein is involved in regulating cell division. And then they look at this protein called PRB, which is a protein which is supposed to regulate cell division. So the kinase puts a phosphate on this one, and then this one is directly proportional to cell division. At least this is what people believe. Now, if you look at the mean distance between cells, 5 minutes after you apply the pressure, it decreases. And it doesn't decrease uniformly in the aggregate. It decreases at the center of the aggregate. So after 5 minutes after applying the pressure, all the cells in the center have a smaller volume, and then this propagates outwards. If you look at the other ones, like P27, nothing happens until almost 1 day. And after 1 day or so, the concentration of P27 increases. It also increases from the center, and you see a wave going outside of P27. And then if you look 4 days after you put the pressure, you see the cell division rate changing. It first decreases in the center, and then there is a wave that moves outside. Now, why does the volume decrease on the center? I don't know, but if you believe John, and I would have said the same thing, there's a flow from outside to inside, so the pressure is larger on the outside than inside. So if you're very naive, you would say it should decrease in the external parts. That's not what's seen. What is seen is that the volume decreases in the center. Now, one of the things that seems to be true experimentally, they then started to worry about the axis of cell division. Close to the edge, the cells divide parallel to the surface. If you look inside, they divide radially. And you could say it's my coupling with the flow goes radially, and that orients the cell divisions. Maybe this counts, but I don't know more than that. So the thing that we want to focus on now is this, because this is kind of the physical part of the problem, but we don't have any idea why it works like this. So let me go to tissue invasion. So I will use the ideas that I told you. The tissues are liquids, and then they have this pressure. The cell division rate decreases with pressure, and the cell death rate increases with pressure. So the problem that I'm interested in is I take 2 tissues, which don't have the same properties. I put it in a box, and they can exchange whatever they want to exchange with the exterior. They exchange water, they exchange nutrients, they exchange growth factors, everything. And then what I want to see is which tissue invades the other one. So until the cells are not confluent, nothing happens. And then you reach the homeostatic pressure of this tissue, which is the one with the lowest homeostatic pressure. So when I'm optimistic, I call it the healthy tissue. And at this point, this tissue is at equilibrium. But this one has a higher homeostatic pressure, so it still wants to divide. So it divides, and it pushes this one inwards. But then the pressure in this one increases, and this one wants to die. So there is no steady state in this problem. The interface moves to one side. And the one which wins is always the one with the highest pressure. It doesn't depend at all on the speed of cell division or anything. The only thing that counts is the value of the homeostatic pressure. Now, for what I want to say afterwards, there is another thing you can do. Instead of using a flat geometry like this, you could use a curved geometry. So you take a small ball of the tissue with the highest pressure. You make the same reasoning, but this time you have to include the Laplace pressure because there is a surface tension between the two tissues. And of course, the criterion is that the highest homeostatic pressure has to be larger than the lowest one plus the Laplace pressure. Or if you invert this, it means that there is a critical radius, like in nucleation phenomena, which is a ratio between the surface tension and the difference is the homeostatic pressure. If the radius is smaller than that, the Laplace pressure is very high, and the tissue inside cannot expand and it dies. If the radius is larger than this, the Laplace pressure is very small. The one inside has a very large pressure and it grows. So that's what we wanted to study more quantitatively. So I will go on with my liquid story. There are two tissues, so there are two conservation laws. And the way we characterize locally the composition of the tissue is by the volume fraction. So the average velocity, average with the volume fraction, is just the average growth rate, where this is cell division minus cell death for the A tissue, and this is cell division minus cell death for the B tissue. Then, if you write the second conservation law, you get the evolution of the volume fraction. So this is a total time derivative. There is one term here which is due to the fact that the tissues don't grow at the same speed. So if this one grows faster, the fraction of A cells increases with respect to B cells. And then there is a relative flux between the two tissues. Now, we studied this relative flux pretty much in details, and it turns out that it's more or less diffusion. So there is a relative diffusion constant between the two tissues that we inject here. And then in order to do things, I will assume that close to the homeostatic pressure, I can just linearize the rates. So that's conservation laws. Then I need to write force balance. Now, because there are experiments in the labs that started with that, what we did is we did it on 2 dimensions on the substrate. So the force balance is just that the gradient of the stress is proportional, is the friction force on the substrate, and we use a viscous friction law. Then you need to know the stress. So there is a pressure, which is the cell pressure. There is a velocity gradient, because I consider it as a liquid. And then there is a coupling to phi here. Now, the most general coupling to phi is what I call the Ericsson stress, because you guys work on liquid crystals. But if you take a phi-4 theory, you get a stress like this. In principle, these two coefficients could be independent, because there is no constraint. At equilibrium, they're related by a factor of 1/3 here. It's proportional to the square of the gradient of the concentration. Now, what this term does is that it creates a surface tension. This term doesn't vanish if the gradient is large, so it doesn't vanish at the gradient at the interface between the two phases. So if you integrate between both sides of the interface, you generate the surface tension like this. And this B here up to a length scale is the surface tension. So the problem that I want to study is this one. I put an A tissue on the left, a B tissue on the right, and this tissue has a higher homeostatic pressure than this one. So it will invade, it will move in this direction, and what you expect is to have a wave that moves in this direction. Now I took the two previous equations, I just dimensionalized them, and I get an equation for the velocity in the tissue, dimensionalized by that, and this is my conservation law. Now if you forget about this term, this equation is extremely well known. It's the Fisher-Kolmogorov equation. So this has a wave that propagates in this direction, and if you forget about this term in my dimensionless unit, the velocity of propagation is 2, which is a classical result for the Fisher-Kolmogorov equation. Now, of course, I want to couple that to convection, to the mechanical effects, and you can view that as an extra nonlinear contribution to this Fisher equation. Now, it turns out, and of course I didn't know anything about it, but that this type of equation is extremely well understood. and that there are general criteria to decide what the velocity should be. So the mechanical effect is just this V naught here, which is proportional to the difference in homeostatic pressure. That creates a velocity, and the velocity interferes with the Fisher equation. So the first thing you can do is you solve it numerically. Now the concentration profile goes from 1, that's where I only have A cells on the left-hand side, to 0, and it has some kind of sigmoidal shape like this, and the shape doesn't depend very much on the parameters. Here the surface tension is 0, but if you change the surface tension, it doesn't change very much. The velocity decays to 0 at infinity on both sides, and there is a length scale of decay of the velocity, which is a parameter that I call lambda. Here lambda is very large, so the velocity is essentially constant throughout the interface. Here lambda is very small, so the velocity is peaked in the middle here. Now if you want to deal with this equation, there is a very nice review written by Vansalos, and what he explains to you is that there are two types of fronts that he called push front and pull fronts. Now the pull front is the easy one. It's the one where the driving by the velocity is very small, and in this case the only thing you need to know is what happens very far ahead here. And in almost all cases, if you start from sharp boundary conditions, it travels at the velocity which is 2 in my dimensionless unit. So whatever V naught, if V naught is small enough, the velocity is 2. If you increase the driving force, then you reach a push front, where what counts now is the term, the convection term, and what fixes the velocity is what happened in this direction here. You have to do some kind of study of the stability of the fixed points of my equation. What you find is a velocity that goes like this, so there should be a small region where it's constant, and then if you go away from this small region, the velocity is just a driving force V naught. So I have two regimes, a Fisher regime where the velocity is 2, which turns out to be very small here, and then a regime where the velocity is just a driving force V naught. What this means is that for this problem I have a competition between two effects. I have a competition between cell division and mechanical properties, and the mechanical effects always dominate. Now if you go back to the original units, the velocity of the front is just a pressure difference divided by the square root of the friction and the viscosity. So essentially the result is that you could forget about the difference in cell division rate. The pressure difference dominates, and that pushes the front, which is what I told you from my qualitative argument. The only thing I have extra is the velocity of the front. Now we did something else. We looked at a circular geometry. So in this case, I take the A cells, I make a small circle of A cells, they have a higher pressure, and I ask when do they invade the B cells. Of course, I take a radius which is smaller than the radius RC. I have something extra compared to what I was telling you earlier, which is that I have diffusion in this problem. So now I have noise. Now if you look at this plot, which is a little bit complicated, this is the profile of A cells in the beginning. The radius of the A cell is smaller than the critical radius. So this number decreases, and as there is diffusion here, the width of the A cell increases a bit. So the critical radius decreases. At some point, diffusion is important enough, the radius has become large enough that the radius becomes equal to the critical radius, and then the number of A cells goes back. Now maybe it's easier to see on this plot. You start from a given number of A cells. In the beginning, the number decreases, and then it increases back. Now, of course, it increases back if you don't reach 0 here, or if you reach something which is larger than 1, and there is a criterion on the surface tension. So even if there is a surface tension, if the surface tension is not too large, because of noise, you can jump over the barrier created by the surface tension, and you can grow the A cells into the B cells. Now what it means, if you want to talk in terms of cancer effects, is that if you have a small metastase, if it's too small, it never grows. But if it's a little bit larger, it can grow by fluctuations. And then if it can grow, it invades, and it can become a large tumor in there. So that's where we are with these things. And as I was saying, there are experiments that have been started, but we don't have any results yet. So that's kind of my story on invasion of a tissue by another one. What I want to tell you shortly now is tell you about villi in the intestine. So your intestine is something that looks like it. It has two parts, the big intestine or the colon and the small intestine, which is connected to the stomach. The small intestine is a tube with an inner diameter of a few centimeters. And if you look at its inner surface, it's not flat. It has protrusion towards the interior, which are called villi, which are this stuff here. And it has holes in the middle of the villi that people call crypts. If you now take a villi and you make a cut, this villi is a monolayer of cells. So this is an epithelial monolayer of cells. And inside here, you have something that people call the stroma. So this is mostly extracellular matrix plus a few cells. Now, if I look at even more details, the tip of the villi would be somewhere here. This is the bottom of the crypts. And people know an awful lot on these systems. Now, this is why we went to that, because there are several biology groups at Institut Curie that work on that. What they tell you is that there are stem cells, and the stem cells are in this region here. I won't detail that more. They divide. If they divide, they push the cells here, so there is a flow of cells from this point to this point, and the cells die at the tip. So that's a very fast story of the intestine. Now, if you look at this structure, so I plot here the surface of the intestine that I make flat. This is a stroma, and for now we treat the stroma as elastic. It's extracellular matrix. There is a basement membrane here, which is a membrane with a higher modulus, and this is my monolayer of cells. Now, qualitatively, what happens is that the cells in the monolayer divide, so they push outwards because of this pressure that I was talking about. So it's as if you were taking this sheet of cell and pushing it inwards, and then it buckles. Now, if you do it with a surface like this, it buckles at 0 wave number. But if you watch in Landau-Lifshitz at the right page, there is an exercise that tells you if you buckle on an elastic medium, you buckle at a finite wavelength. So all this story here is to tell you that there is a buckling. There is a well-defined wavelength for buckling that depends on the bending energy of this membrane here and on the elasticity of the substrate, which is here. If you put numbers, you find something like 100 microns, which is a reasonable value for most animals you can think of. So the mechanism that we have in mind is that because of the growth of the cell, it buckles. Now, how do we reach that? Essentially, as before, we write a conservation law for the cells. It's cell division minus cell death. Cell division depends on the pressure, and the pressure is minus the tension times the thickness. So I use the tension here. This is the homeostatic value. And I added another term here, which is the fact that this interface is polar. It's not the same on both sides. So if it's not the same on both sides, the division rate can be coupled to curvature. So there is a term which is linearly coupled to curvature in the division rate because the interface is polar. Now, what this tells you is that if you choose the sign of this in the right way, cell divides when the curvature is like this, and they die when the curvature is like this, which is what biologists tell you. So essentially, I have two parameters, the tension here and this asymmetry parameter here. Now, you can do a nonlinear theory of buckling, and people in mechanics know how to do that very well. It's called the Foppel-von Karman theory. So we used that, and we calculated the shape of the villi. This picture here is made when there is we ignore the coupling between cell division and curvature. So what you have is villi here, you have crypts here, and as I ignore the coupling, everything is symmetric, so the villi and the crypts are perfectly symmetric. And if you look carefully, it makes a square lattice. That was known. If you take a metallic sheet, you put it on an elastic medium, you push it isotropically and it makes a square lattice. That was calculated a few years ago by Yareski Boudaoud and somebody else in Paris.
But it's only the elastic sheet that's under
So,
stress.
so it's what's on the sheet is just the layer of cells, and then there is a friction with a substrate below.
Right, but the substrate is not under any stress.
The substrate is coupled to it by the friction.
Right, but it's not, you're not.
There is no stress. I'm not pushing on the substrate. I see that I will have to stop. Let me tell you about this one. So I increase the asymmetry. So I increase the parameter which is coupling curvature and cell division. Of course, if I increase this parameter, I increase cell death at the top here. So the villi are just cut. So there are only crypts and there are no villi. That's kind of obvious. The thing which is not obvious is that you go from a square lattice to a hexagonal lattice. And it turns out that this is actually what is observed. If you go in the small intestine, and this is a picture of the small intestine, you have crypts and villi. If you go in the colon, you only have crypts and there is no protrusion outwards. So what I would say is that the coupling to curvature is much higher in the colon than it is in the small intestine. Now, if you look at the structure of the villi in the small intestine, it's a hexagonal, it's a square lattice, and this is what was on the previous picture here. 1, 2, 3, 4. And if you look in the colon, you have a hexagonal lattice. Now, I will stop here. Just before that, I will mention that there are other kind of structures that you can find. Instead of having fingers, you can have ridges, and you find phases where you have herringbone structures, and you find other stuff which is a random labyrinth phase. These also exist in the intestine of small animals. They exist in the region where the activity of the cell is very high, which is close to the stomach. That perfectly corresponds to the theory. So I think Len wants me to stop. So I will stop here and thank you for your attention. Yeah. Peter.
Questions to this moment. Or yeah, please, but. To see square to hexagonal, you need nonlinear terms. There are
So
nonlinear terms.
the nonlinear terms are in the front. Okay, I can show you where they are. I didn't write them explicitly. In the Foppel-von theory, the nonlinear terms come from the nonlinear elasticity of the membrane. We also kept the nonlinear geometry, in the nonlinear geometric term, which is there is one in the bending modulus, there's one in the negative tension.
So there's obviously a lattice constant in your square and hexagonal lattice.
Yes.
Can you somehow get
So
a simple relation between your parameters and this?
So
lattice constant?
what is simple is you do a linear stability analysis, and you find that, and you get something which is close to the lattice constant. What you find is that it depends only on the bending modulus of the basement membrane and on the elasticity of the substrate, which means it only depends on material properties of the intestine. Then it means that any animal you should take, the period should be reasonably the same. And that's true. If you take a man or a mouse, the period of the villi is not very different. The period of the villi is of the same order of magnitude as the amplitude of the villi. So it's a couple of 100 microns.
Could you point out what sets the length scale for these structures, these
So
square
that's
arrays?
what I was saying. What fixes the length scale is the ratio of the elasticity of the basement membrane to the elastic modulus of the substrate to the 1/3 power. The 1/3 power is very nice because if you make a mistake by a factor of 10, you see it gets the right number. So it allows you to make almost quantitative comparison, even if your parameters are not so well known. No, you need it.
So regarding your observation on the increased death rate in your balls of
Yes.
cancer cells, I was wondering whether there might be any role played by dehydration of the cells due to the extra osmotic pressure.
No, so that you can estimate. You can say there's a gradient of chemical potential of water. How much would the volume of the change of one cell change with that? That's completely negligible. It's much less than what's measured here. We thought about that.
Yeah, so everything does depend on alpha having the right sign. Is there an independent way of estimating it or anything?
No, but there's nothing so special with the intestine here. So you can say there are other tissues that will do the same thing, and the skin can do the same thing. And so there's something called papillae of the skin, or you can think of your fingerprints. Now in the skin, apparently the stem cells are on top, and the structure is inverted. So it's only the absolute value of alpha that counts. The sign just tells you where the dividing cells are, and they can be either on top or on the bottom.
Okay, now the last question, can you come up, please?
So if you, first thing is, I wonder what's the role, if you have the fissure, I guess the fluctuations play an important role on the tip of the front. And the second part of the question is, if you have a benign tumor, I guess that the surface is relatively smooth, but otherwise, I get this is quite rough, and you have metastasis and so on.
So what you're saying, so the Fisher equation, the only noise that I put is the diffusion constant here. I agree that if you put noise on the Fisher equation, the velocity of the front depends on the noise.
Cells, right?
Sorry?
You have the proliferation of the cells. You have
Proliferation
quite
of
a
the cells has
stochastic
noise.
activity on the
We
front.
try to estimate that the effect of the noise didn't seem to be very big. But I know that this is well understood. Derrida, for instance, had done serious work on that. It's not included in what I showed. Now, for the benign tumors, the view that I have that might be wrong is that the benign tumors, they are surrounded by what I call the basement membrane. Now, if you have a basement membrane, it can grow, and if the membrane is elastic, you can have a stable steady state, and that's a benign tumor. Now, there are all kinds of biological processes which are such that the cancerous cells which are close to the membrane start to attack the membrane. Then, if you make a hole in the membrane, they start to leak, and they can go and metastase somewhere else. But as long as everything is bounded by this basement membrane, the cancer is not, quote unquote, dangerous because it doesn't metastase. So the view that we have is that what people, in our language, what a benign tumor would be, is a cell aggregate that reaches equilibrium. Now, if you only have a tension, as I said, there is no equilibrium state. I mean, there's one, but it's unstable. If you make the membrane elastic, you can reach equilibrium state. And you see, I mean, if you look on the web, you see benign tumors which are enormous that people got extracted.
Well, let us speak.
Yes.